Hello, everyone. My name is Yuki Kato Maves, and I lead the product management team for the biospecimens products and services business of Discovery Life Sciences. And today I will be discussing from a starting cellular materials perspective, how we can address the challenges of allogeneic GMP manufacturing using healthy donor PBMC isolation as an example. I'll start with a brief introduction to Discovery Life Sciences and explain how All Cells plays a crucial role within the broader context of our organization and for the cell and gene therapy industry. Then I'll talk about current trends and challenges in cell therapy manufacturing, and I'll demonstrate how, as a provider of healthy donor cells, we can help further mitigate risks in bringing allogeneic cell therapies to market using a real-world case study. Then lastly, I'll offer a sneak peek into our new product, the GMP cryopreserved PBMCs. So Discovery Life Sciences is a leading global provider of biospecimen products and specialty lab services. We enable the discovery and development of novel therapies and diagnostics by providing companies with the highest quality patient biospecimens, in vitro preclinical solutions, and cell and gene therapy starting cellular materials. Additionally, Discovery offers a comprehensive suite of specialty lab services, including molecular pathology, genomics, cell biology, and proteomics to support both prospective and retrospective clinical trial analysis. All Cells was acquired by Discovery Life Sciences back in 2022 and is now the cell and gene therapy brand of Discovery. Healthy donor cells is at the core of what we offer, and we can support a wide range of projects from basic research all the way through to commercialization. Our collection facilities are wholly owned and operated, meaning that we have full control of the donor supply chain. And as we have tailored a unique donor ecosystem with a large core of well-characterized, highly engaged, and recallable donors, which is how we uphold a 98% collection deliverability rate. We also have an ISO 7 GMP clean room co-located at the Alameda, California collection facility. And to date, we have shipped over 600 GMP collections in order to support process qualification activities and clinical and commercial GMP manufacturing. And here's an overview of the All Cells primary human cells products. We have a variety of whole tissues for research use, from mobilized to non-mobilized leukopaks, bone marrow, cord blood, whole blood, and isolated cells from each of these tissues. And we have RUO to GMP continuity for fresh and cryopreserved leukopaks, mobilized leukopaks, and bone marrow. And I'm excited to announce that our first isolated GMP product will be coming online shortly, which is the GMP cryopreserved PBMCs. So looking at the current landscape in cell therapy, some notable trends include increased efficacy in treating blood cancers, with several autologous CAR T cell therapies gaining earlier line treatment approvals. Progress is also being made against solid tumors, and there have been clinical breakthroughs in treating autoimmune diseases with cell therapy. And while very exciting and promising, these new trends do highlight new and pre-existing challenges, such as a much larger patient population with not only cancer indications, but prevalent autoimmune indications as well. Securing reimbursement from payers continues to be a challenge, and there's still limited broader adoption due to the complex logistics and high costs. And then increasing and evolving regulatory requirements emphasize raw material compliance as well as process robustness. So it's evident that continued advancements in technology, supportive regulatory frameworks, innovative reimbursement models, and strategic collaborations are going to be the key to overcoming these types of challenges and driving the growth of the overall cell therapy market. Because patients in dire need are the ones suffering if we as an industry don't address these challenges. And the statistics shown here are a stark reminder of the cost of failure. 4 to 7% of patients are unable to receive their CAR T cell products due to manufacturing failures. 20% of myeloma patients will be unable to receive their cell therapy due to the long wait time. The high cost of CAR T makes payers unwilling to pay for these life-saving treatments, and patients like Forrest Van Patten here are the ones that are paying the price. High COGS and manufacturing failure rates drive up therapeutic pricing, complicates reimbursement, which limits patient access and reduces the overall commercial viability of cell therapies. And so we know that we must do better to make these therapies more cost efficient and more widely accessible. Now, patient accessibility is an incredibly complex topic, but some of the most critical factors in order to improve patient accessibility is by controlling costs and improving manufacturability and scalability, which have direct implications on drug prices as well. And there have been significant effort and innovations from across the industry to address this. And the allogeneic and cryopreservation approaches have become a driving trend because these enable economies of scale. And in fact, the Alliance for Regenerative Medicine Sector Snapshot reported that allogeneic approaches represent about half of phase 1 cell therapy trials with known approaches. And at Discovery, we also want to do our part to reduce the costs as well. And we do this by supporting scalable allogeneic approaches, by delivering high quality starting cellular materials sourced from the right healthy donors, delivered when they're needed with great reliability. We also have optimized cryopreserved starting materials, which maintain quality while extending shelf life and overall streamlining logistics. We are cell and gene therapy focused and client-centric, meaning that if we don't currently have what you're looking for, we will work with you to provide customized solutions through our product development and validation capabilities. But even for allogeneic approaches, there are opportunities to further de-risk the workflow. And if we think about a typical process where we send a healthy donor fresh leukapheresis material from a collection facility to the manufacturing facility for target cell isolation and further processing, we can already identify multiple potential points of failure. One being that if there is a delay in processing, this can introduce poor starting material quality, which introduces process variability, and in the worst case scenario, batch failures. And then with fresh products, you are inevitably vulnerable to logistical disruptions. And then inefficiencies in labor and resource utilization across the workflow can exacerbate the already high costs as well. And so, how can we de risk this, right? One of the ways is, of course, to leverage cryopreservation, and cryopreserved starting materials are being explored by many programs. We can also go directly from apheresis to target cell isolation under one roof and collapse that workflow. This can further streamline the process and reduce logistical challenges and cost as well. So, with that concept in mind, today I want to share a real life case study in which a key client of ours pioneered a solution for their manufacturing bottlenecks by leaning into Discovery's capabilities. So, this particular client is in the process development stage for their allogeneic cell therapy candidate. And they utilize the traditional model of procuring a fresh leukapheresis material from a collection facility, which gets flown to their CDMO and is subsequently processed into PBMCs. The CDMO uses an open process to isolate, and they use an in-house cryopreservation media to cryopreserve their PBMCs. And here are the challenges that they are facing. So the delay in processing the fresh leukapheresis material was impacting their cell quality. And they are also unable to predict the cell performance up front. And this was leading to a higher than desired failure rate of their manufactured drug products. And lastly, the cost of utilizing a CDMO suite for this relatively simple step was just another additional cost to them. And so, in order to solve their specific manufacturing challenges, here were the requirements that we needed to meet. They wanted the ability to pre-screen the PBMC batches so that they could conduct a pilot run in-house to predict cell performance in a smaller scale and to only select the lots that worked for them. They also wanted to improve their manufacturing suite utilization strategy, and they needed a partner that could deliver consistent GMP compliant healthy donor PBMCs. So that meant having a robust quality management system, an ISO 7 clean room, and GMP compliance, FDA and EMA compliance for their regulatory jurisdictions, donor and GTP collection expertise, and not just a tech transfer, but the infrastructure and collaborative team to support product development and validation capabilities. So ultimately, the client screened many different providers, and Discovery was chosen to collaborate due to our unique positioning. So essentially, by doing the isolation and cryopreservation at the leukopak collection facility, they're able to leverage the full control of the donor supply chain to yield high quality isolated cells. They would be able to minimize the cost of failure by predicting cell performance upfront and only selecting those preferred lots. And they would be able to optimize their CDMO utilization and enable the CDMO to focus on complex downstream manipulations. So our team is currently knee-deep in validating the process, but preliminary results show that the new process and workflow has improved the cell quality significantly, with post-thaw viability of the isolated PBMCs going from 50% to over 90%. And then on the right here is a high-level estimate of the cost savings going from a 60% manufacturing success rate to 100% to achieve a hypothetical 10 past manufactured lots. This takes into account extensive allogeneic donor screening, leukopak procurement, cell isolation, and manufacturing. And essentially, you need 17 collections and manufacturing runs to end up with 10 pass lots at a 60% pass rate versus just doing the 10 runs. And I understand this is somewhat simplified, but in this scenario, our client would be projected to save roughly over $50,000 per run. And now for others, the economic value of this process might vary depending on the manufacturing success rate, amongst other things. For example, do you already have an isolation process established, or is that something you have yet to do? Do you have a facility and personnel trained? And so it really differs from program to program. And so we understand that it's not a one-size-fits-all calculation. There has been a lot of progress made to date, including aligning on the product development terms and deliverables, having a robust communication plan with working and governance teams to really enable timely and efficient decision making. We've had regulatory discussions with the appropriate regulatory body. We have completed the process development runs from which the samples were tested by our client, and they were very happy with those. And we are well underway to qualify the process and bring this into full production by the end of this year. So I think this case study really highlights the important role of collaborations in order to drive innovation and accelerate cell and gene therapy development. These types of partnerships reduce costs and risks, enhance capabilities, and improve quality and efficiency. Because at the end of the day, we must remember that we are all working towards a common goal, which is to accelerate the delivery of novel treatments to patients in need. And so together with our client, we both realize that this workflow could benefit other programs. And our client is more than happy to make this process available to other stakeholders once it's established and validated, to help more programs get to the clinic and accelerate approvals. So due to the nature of the collaboration and our client's pre-screening paradigm, we'll have two main types of GMP cryopreserved PBMCs available, one being off the shelf and the other being custom made to order. Now, what will be universal is that the donor will meet the 21 CFR 1271 criteria at the very minimum. The isolation process itself will be in a closed system, and the cells will be cryopreserved in the industry gold standard cryostore media. There will be viability, cell concentration, and volume specifications, and the product will be safety tested for sterility and endotoxin. Off the shelf means it's going to be ready to ship with a predefined set of specifications. And these will be available for pre-screening, as I described earlier. So you only select the batch that will maximize your downstream manufacturing success for your particular program before committing to purchase of either the rest of the lots or just more vials from that lot. And the beauty of this is that you can also save time on paperwork like the SOW and MSAs. Custom made to order is exactly what it sounds like. We can tailor to the program specific donor criteria and the infectious disease marker testing, and you'll be able to choose whether this should be in a vial or a bag format. And now there are some nuances to this, so I encourage you to reach out for more details if you are at all interested in learning more. Dynamic, constantly evolving, these are the phrases that really come to mind when thinking about the cell and gene therapy industry. And we are doing the same as we identify unmet needs and evolve in parallel with the industry to work towards decreasing COGS and timelines. And so the GMP cryopreserved PBMCs that I just described will be ready in Q1 of 2025. And then we are also looking at enriched cell subpopulations that can further reduce steps in the workflow for various types of therapies, including T and NK-based therapies to engineered hematopoietic stem cells, feeders for TILs, what have you, to make things easier from both a logistics and a cost perspective, and really doing our part to remove bottlenecks and to collapse timelines. I really appreciate your time and attention today. Of course, please reach out to us if you have any questions or would like to learn more about the new GMP cryopreserved PBMC product. Also, I encourage you to reach out to us to discuss opportunities to de-risk your manufacturing workflow or improve your process scalability. So thank you.